Okay. All right. Okay, we'll make a start. So the basic immunology that I told you about in the POD block is essentially, if you imagine like a metro railway system, it's a central line, and it's more of an induction type mechanism because the lymphatics and the blood is essentially a sterile environment. So rarely do you get antigens, foreign antigens invading in there unless it's, say, a cut in the skin or a insect bite or whatever. Where the immune system is most active is in the barriers of the body, like particularly, well, in the mucosal system, the respiratory tract, the GI tract, the skin, urogenital tract, and so on. This is where most of the immune cells are located, and this is where they are most active. So they have a different function, and also they are more tightly regulated compared to what you taught in the secondary lymphoids, lymph nodes, and so on. So the objective of this lecture is to describe the composition of this so-called gut-associated lymphoid tissue, or GALT, and to describe the types of cells and their function that make up this mucosal immune system. These cells, just like the cells that you learned that circulate in the lymph and the blood, most of these cells originate from the thymus. But some of these cells are also specialized within these mucosal barriers. With regard to antibodies, the primary antibody is IgA, and I'll talk about the properties of IgA. But bear in mind that IgG, possibly IgE, also function within the system. And then an important component of regulation, what you call education of this type of immune system, is the microflora, the bacteria, particularly bacteria in priming and educating the immune system what to react to and what not to react to. So first of all, with regard to the unique structures, immune structures associated with the gut, we first have this system of lymphoid organs lining the oral cavity, what's known as Voldier's ring. This includes the adenoids and the tonsils, which are essentially filled with B cells, because this is the first passageway through the airways and the gut where you encounter foreign microbes, aerosol antigens that you either ingest or breathe in. And then in the intestine, in the gut, you have a whole system of very fine lymphoid lymph nodes. These are the mesenteric lymph nodes. And these are linked to those lymph nodes that line through the body. Lining the small and the large intestine are these specialized lymph nodes known as Peyer's patches. These contain your classical B cell follicles, T cell zones, dendritic cells, and so on. And then at the end of the gut, you have the appendix, which is another specialized structure. And this is filled with microbes. So if we do a cross section of the small intestine, you have, as you learned already, these enterocytes, which are primarily involved with digestion, absorption of nutrients from food. This is the lumen of the gut. Under in the underlying tissue here, in the basement membrane, you have the lamina propria, which is filled with the usual immune cells like NK cells, T cells, T helper cells, B cells, and so on. This lamina propria is circulating from the lamina propria are lymphatics which go to the mesenteric lymph nodes. And this is where any T cells which or B cells which get activated here will move into the through the lymphatics to the mesenteric lymph nodes for further differentiation and seeing antigen from dendritic cells and so on. The also important feature too are isolated lymphoid follicles, which are individual lymphoid follicles containing mostly B cells. And these function just like the lymph nodes in the general systemic system. But these lymph nodes don't require T cell help. They predominantly produce IgA. And then also an important feature of this gut-associated lymphoid system are these T cells, which are within this epithelial barrier, this mucosal barrier. These are known as intraepithelial lymphocytes. So firstly, if we just go back to the basics of innate immunity, the epithelial cells which make up the mucosal barrier, if you just recall your very first POD immunology lecture, as part of the innate immune system, the anatomical features, these epithelial cells themselves also play a role in the immune system by virtue of the fact that they express PRRs such as TLR5, which can recognize PAMPs like flagellin on bacteria that enter the gut lumen. And so once the PRR is engaged, these epithelial cells can produce cytokines to activate other parts of the immune system. An important product of these epithelial cells are these so-called antimicrobial peptides, which are very small, which are peptides which can directly kill any pathogenic bacteria that enter the lumen. So as I said, within the lamina propria, you have your normal whole range of immune cells like mast cells, which can release histamine, induce an inflammatory response, dendritic cells for antigen presentation, macrophages for also antigen presentation, cytokine production, and so on. CD4 T helper cells, CD8 cytotoxic T cells, antibody-producing plasma cells. These intraepithelial cells are between the basement membrane and the tight junctions that make up this epithelial barrier. These aren't just fixed, but they can move through this whole basement membrane and actually play a role in immune surveillance, looking for any infected epithelial cells in that barrier, as in the case of a virus-infected epithelial cell, and kill it through classical cytotoxic T cell mechanisms. But once they've done that, they also have cytokines which repair that tissue to make sure that epithelial barrier is maintained, and so any pathogens which infect don't penetrate and then get into the underlying tissue and maybe into the bloodstream. And then another important cell which is involved in this gut-associated lymphoid tissue are these innate lymphoid cells. Remember, there's ILC1, ILC2, ILC3, which produce cytokines similar to Th1, Th2, Th3. ILC3, its classical signature cytokines, if you remember, is interleukin 17, which is a predominantly inflammatory cytokine directed against fungi, helminths, and also bacteria. But also, it is induced through IL-23. But once these fixed ILC3 cells are activated, they can then act to kill off any pathogens which invade this epithelial barrier. But they also induce these cells to produce antimicrobial peptides to aid in this protection against infection. And also, they produce IL-22, which acts to repair and replace any of these damaged epithelial cells to maintain this barrier. The intraepithelial lymphocytes actually make up most of the lymphocytes in the body. And this is because the small intestine and the large intestine makes up such a large surface area. So there are much more lymphocytes that survey this whole area compared to what's circulating in the lymph and the blood. There are many types of these intraepithelial lymphocytes. Most of them are CD8 positive cytotoxic T cells, which contain the alpha beta T cell receptor. But some of them also contain unusual types of cytotoxic T cells where they only have the alpha chain. So they are double alpha-alpha T cell receptors. And then you also have some that have the gamma delta T cell receptor. So they kill, if they see an infected epithelial cell, they will kill it through recognizing antigen presented on class 1 MHC and kill that epithelial cell through granzyme perforin mechanisms, just like in the normal CTLs. But they can also kill these infected cells through Fas ligand interaction. But another feature of these cells is that once they've killed these cells, they can also produce cytokines which replace or induce a new epithelial cell to maintain that barrier. These intraepithelial cells originate from the circulating lymph, but once through various signals, they change their chemokine receptor expression such that they no longer circulate through the body lymphatics going from lymph node to another, but they will then be directly homed to the gut tissue and also into the epithelial barrier. And this is through expression of CCR9, this chemokine receptor, and this integrin alpha E beta 7. With regard to the adaptive immune response, first of all, this specialized structure, the Peyer's patches. And so these are specialized lymph nodes scattered throughout the epithelial barrier, which, just like a normal lymph node, contains dendritic cells, B cell zones, and T cell areas. And they sample antigen, which is in the lumen of the gut, which passes through into this capsule where they are taken up by dendritic cells, and the dendritic cells can then present and activate T cells and so on. But also, there's another specialized epithelial cell scattered throughout the barrier membrane amongst these enterocytes. And these are specialized enterocytes known as M cells or microfold cells. Instead of expressing villi for nutrient absorption, they actually have all these folds which through receptors can bind to any antigens that are in the gut lumen. And through transcytosis, these antigens pass from the luminal side to the basolateral surface into the Peyer's patches for presentation by these dendritic cells. So this is how these M cells take up the antigen, that they can either just through pinocytosis and endocytosis take up these antigens into vacuoles, or they can and then transport it where it is then taken up by these antigen presenting cells. Another type of capture of antigens in the lumen of the gut, there are many mechanisms whereby these antigens can be taken up. One is through the transcytosis through the M cell, like I said. The other is through the capture by these receptors, such as the neonatal FC receptor. If you recall, I talked about neonatal FC receptor before. The neonatal FC receptor is a receptor for IgG. It allows for IgG in a pregnant mother to cross the placenta to protect the fetus. It is also responsible for it is also the reason why IgG is most viable compared to other antibody isotypes in the blood, because IgG by binding to the neonatal FC receptor, can transcytose through cells and not be degraded within lysosomes and then exit out. And so this is why it is prolonged. So in this setting, this neonatal FC receptor also functions to transcytose IgG, which is in the lumen of the gut, and that has captured any, say, bacterial antigens and then passed down into the basolateral surface, and then that antigen then gets captured by dendritic cells for antigen presentation within this Peyer's patch. Another mechanism for antigen capture is that any epithelial cell that is part of this barrier that gets killed, as in the case of a virus-infected epithelial cell and undergoes apoptosis, these apoptotic bodies can get taken up by the dendritic cell. And then a fourth way of sampling antigens in the gut lumen is that macrophages can extend processes through the tight junctions into the lumen where they can capture antigen and then bring it down into the basolateral surface for antigen presentation. All of this is to sample any antigens in the gut and then give it to these responder T helper cells. This is not only to activate the immune response to react to these antigens, because the antigens which enter the lumen of the gut are both potentially pathogenic, but most of them need to be tolerized to the immune system, because most of these antigens are innocuous food antigens that we are constantly taking up. So this is where this mucosal immune system needs to be educated to be much more discriminatory into deciding what should I react to and what should I leave alone. So this is an important part of peripheral tolerance, because any breakdown in this mechanism can lead to inflammatory bowel disease, Crohn's disease, and so on. So although I said that the neonatal FC receptor can transcytose IgG, IgA is the predominant antibody that is associated with the mucosal immune system. And as I said in the POD, IgA, like IgM, can form multimers, but with IgA, these are essentially dimers. So you can get two IgA antibodies that are linked via the FC receptor through a peptide known as the J chain or joining chain. So this then, having two IgA molecules, you have 4 binding sites through the FAB, but it also allows for that dimer, that dimeric IgA, to cover a much wider surface area. The IgA is the largest amount of antibody produced in the body. But if you look in the blood, the predominant antibody, as you know, is IgG. So even though the body produces the most abundant, IgA is the most abundant antibody, most of the IgA is secreted from the body in terms of the gut, in terms of excretion, or in breast milk, in sweat, tears, and so on. And this reflects the main function of IgA is not to induce an inflammatory response which would result in the death of cells and the breakage in this mucosal barrier, but to bind to any potential pathogenic antigens and to excrete that antigen through sneezing, coughing, or diarrhea or vomiting. The IgA isotype consists of two types, IgA1 and IgA2. These differ with regard to the FAB sequences, where IgA has a longer hinge region that allows it to cover a wider surface area. One of the what do you call it, for bacterial forgotten, the factors that bacteria make to make them more pathogenic or survive, what's it called in microbiology? Yeah, all right. So some bacteria like Haemophilus and Neisseria gonorrhea make enzymes, these virulence factors, which can cut the IgA and so make that IgA1 ineffective. IgA2, the FAB region of IgA2 is resistant to these virulence factors. So this is a backup system for countering these virulence factors made by these bacteria. So again, just like IgG, IgA can transcytose from the basolateral part of the epithelial barrier to the luminal side. And it does it through this other receptor known as the poly-Ig receptor. So remember, you have the neonatal Fc receptor for IgG, you have the poly-Ig receptor for IgA. This poly-Ig receptor for IgA binds via this J chain and then passes the dimeric IgA into the luminal surface. And this J chain gets cut, and a portion of the J chain still remains to link up both IgA monomers. And this J chain can anchor itself into the lumen of the gut, where this dimeric IgA can then sample any antigens. And if it's a pathogenic bacteria, this can get flushed out of the system. Otherwise, this dimeric IgA can then transcytose back down to the basolateral surface for antigen presentation. So this shows what I've just said, that there are many functions of the dimeric antigen. Firstly, if there is invasion with a pathogenic bacteria that produces toxins that penetrate into the basolateral surface, this dimeric IgA can capture these toxin molecules and then transcytose into the lumen, where they can then excrete that toxin. Going from the other way, if the toxins or microbes penetrate the epithelial cells, as the dimeric IgA is transcytosed, it can capture any of these antigens within the epithelial cytoplasm and then excreted into the lumen. The other function of the thymeric IgA, as I said, if it is anchored within the lumen, it can capture any of these toxins to allow for their excretion. And with regard to antigen capture and presentation, any bacteria that are in the lumen get taken up by the dimeric IgA, transcytosed into the basolateral surface, maybe into Peyer's patches, where the antigen can get taken up by dendritic cells for antigen processing and presentation. So the differentiation and maturation of these IgA producing precursor B cells starts in the Peyer's patches, where, if you remember, these naive B cells initially express IgM. Okay, when they encounter antigen and through T cell signaling switch from IgM to the production of IgA, these activated B cells will then pass into the circulating lymph to the mesenteric lymph nodes. In the mesenteric lymph nodes, they undergo further differentiation where they change the expression of chemokine receptors and express alpha 4 beta 7 integrin, CCR, and CCR9. Within the mesenteric lymph nodes, through signaling with the T helper cells, they then pass back into the lymph and then in through the thoracic duct in the blood, circulate through the body, and then home directly back to the mucosa by virtue of the fact that the expression of alpha 4 beta 7 and CCR9 on Th2 cells bind to this addressin MADCAM in the gut tissue and CCL25, which is the reason or the mechanism whereby these activated B and T cells then migrate and become specialized gut immune cells. So the lymphocytes that are primed within this gut immune system are very different from the immune cells which are circulating from one lymph node to another. And this is by virtue of the fact that they're expressing this CCL9 and alpha 4 beta 7 and alpha 4 beta 7. So another the major factor of these immune cells which are involved with the gut is that they need to be a lot more discriminatory about what is pathogenic and non-self that I should attack and induce an inflammatory response, and what should I leave alone, as in food antigens or microbial commensal bacteria that fill up the gut. And so this is part of this peripheral tolerance mechanism. We can demonstrate this by the fact that if we give a mouse regular oral doses of an antigen like ovalbumin, egg albumin. And then after several doses of this oral antigen, we then inject the peritoneum with the ovalbumin with adjuvant. And we find that the mouse doesn't produce antibodies, doesn't produce an activated immune system against ovalbumin. So the conclusion of this is that through oral exposure of this antigen, it allows for tolerogenic mechanisms such that the circulating or the systemic immune system does not react to the antigen. How is this maintained? How is this brought about? This is through the activation of regulatory T cells or Tregs. If you remember, regulatory T cells produce cytokines like IL-10 and TGF-beta, which suppress an active immune response to these antigens. So the mechanism of this oral tolerance is to induce the production and activation of regulatory T cells, which are central to this peripheral tolerance. Another way of inducing tolerance is to give the animal large doses of the antigen. And this other type of tolerance is the result of deletion or energy as a result of too much exposure to that antigen. So these regulatory T cells are induced through the type of antigen presenting cell, namely the dendritic cell, which is the result of various signals that are produced depending on the type of antigen which is seen by the dendritic cell. If you remember with macrophages, I talked about two types of macrophages, the M1 macrophage, which is your classic inflammatory cytokine producing antigen presenting cell, and the M2 macrophage, which is involved in homeostasis of just phagocytosing antigens to remove them from the system. The same thing happens with dendritic cells. We now realize that there are several types of dendritic cells, some which induce this classical inflammatory response, the other which induces the outgrowth and activation of regulatory T cells. So the production of these so-called M2-like dendritic cells is through the production of various signals and chemicals produced by epithelial cells, such as this TSLP or thymic stromal lymphopoietin, TGF-beta, and prostaglandin E2, which can induce this dendritic cell into this M2-like regulatory inducer. This is a classical inflammatory type dendritic cell, which, if you remember, will then capture, present antigen on class 1, class 2 MHC, and express these co-stimulatory molecules of CD28, B7, which activate your classical T helper cells and cytotoxic T cells. With regard to the regulatory T cell inducers, once this Th2 type dendritic cell is expressing IL-10, retinoic acid, which is an important signal to induce a regulatory T cell, this this naive T cell, which gets induced into a regulatory T cell, the signal to induce a naive T cell into a regulatory T cell is this transcription factor, FOXP3. If you remember, when I talked about T helper cells, you have these transcription factors like T-bet, gamma ROR, which induce Th17s, and so on. FOXP3 is the transcription factor which induces regulatory T cells. And we know this because there are children who have mutation such that they cannot produce a functional FOXP3, and so they have no regulatory T cells. These kids are very susceptible to developing inflammatory bowel disease, as well as reacting lethally to allergens like peanut allergies and so on. So this shows the importance of this transcription factor in its role in inducing regulatory T cells. So again, the important point then is that the immune system of the mucosal barriers, and in particularly the gut, needs to be able to discriminate between what is pathogenic and should be dealt with through an inflammatory response and what should be tolerized to. And this is through the type of antigen which it sees, whether it's part of the microflora or whether it is food antigens. And this comes as soon as the fetus is born, as it passes through the birth canal and then gets exposed to any bacteria that start to colonize in the gut and also through mother's milk. The evidence for this comes from the fact that we can develop strains of mice which are born in totally sterile conditions, where they are fed autoclaved food, so they have very little bacteria in their gut. And these animals have very underdeveloped immune system. They have very poor development within their bodies. So this video is just basically a summary of this whole gut immune system. Let me see. There is a narration to this. You can watch it on your own computers. But basically, these are the enterocytes with all the villi, and these are Peyer's patches here. And these are all the antigens in the gut lumen. I think it's better if you watch it afterwards with the narration. Uh, so, in conclusion, then, firstly, you need to be aware about these unique features, anatomical features, and structures, such as the Peyer's patches, the intraepithelial lymphocytes, these isolated B cell follicles that line the gut. You also need to be aware of the mechanism whereby any of these immune cells that are part of this gut immune system, even though they originate from the thymus, when they are activated, they express these particular chemokine receptors where they circulate back into the blood system and the lymphatics and then home directly through the gut by virtue of these chemokine receptors like CCR9, CCR10, and linking or recognizing these addressins like MADCAM1, which localize them to the gut. Then the other important feature you need to know is the function of IgA, that is the predominant antibody within this mucosal immune system, that it is functions mostly to expel any antigens, and then also to realize that the immune system of the gut is far more discriminatory in terms of inducing peripheral tolerance to not react to and induce an inflammatory response to particularly innocuous antigens.